imagine trying to find a single misprinted letter hidden inside a library of a billion books.
Yeah, that sounds like a nightmare.
Right. Now imagine that if you guess the wrong letter, it actually costs a pharmaceutical company $1 billion and 10 years of wasted time.
Exactly. Not to mention the patients.
Yeah, exactly. While you're guessing, millions of people who are suffering from a chronic, debilitating illness are just, well, they're left waiting for a cure that never actually arrives.
It sounds like an impossibly high-stakes scenario, but that is the exact literal reality of modern drug development.
It really is.
Historically, about 90% of drugs just, they fail in clinical trials. And they don't fail because the chemists aren't brilliant.
Right, the chemistry is usually fine.
Right. They fail because the initial biological target, that misprinted letter you mentioned, it was just wrong from the very start.
That failure rate is just, it's staggering. And it's exactly why we are plunging into this deep dive today.
I really need to unpack this one.
We do. So for you listening, we are looking at a highly technical set of notes today. It's a blueprint titled The Complete 2026 Causal Genetics Pipeline.
And this isn't just another standard research paper.
No, not at all. This is a massive, fully automated machine designed to solve that exact needle-in-a-haystack problem.
The whole mission laid out in these notes is about taking this incredibly messy, chaotic ocean of human genetic data,
Which is huge.
well, massive, and ruthlessly distilling it down to high-confidence, scientifically bulletproof drug targets.
It really represents a profound shift in how we do science.
It absolutely does. What the authors have engineered here is a rigorous, completely unforgiving 7-step gauntlet.
A gauntlet is the perfect word for it.
Right. Every single phase, like every algorithm in this pipeline, is designed to just aggressively hunt down and eliminate false positives.
They want to strip away all those statistical illusions.
Exactly. The goal is to leave only the pure causal biological truth.
Okay, so to make this towering stack of algorithms and acronyms concrete for now, we are going to use a running example throughout this deep dive.
I think that's a great idea. It gets very abstract otherwise.
Yeah, we have to ground it. So we are going to track a single disease, rheumatoid arthritis, or RA.
Which is a really complex one.
It is.
Yeah.
For anyone who doesn't know, RA is this brutal autoimmune condition where basically your own immune system decides to attack your joints.
It's exactly the kind of mysterious disease that has historically just stumped researchers.
Right. So we are going to imagine feeding the mystery of RA into the start of this pipeline, and we're going to track it through all 7 steps.
And if we look at the overarching philosophy of this source material, one thing becomes brilliantly clear right away.
What's that?
The individual computational tools we have in genetics today, they're impressive on their own, but they become truly transformative, almost terrifyingly powerful when you chain them together.
Like a unified sequence.
Yes. The ultimate prize this pipeline is hunting for is something researchers call cell type resolution, backed by multi-omic evidence.
Okay, let's unpack this, because that brings up a critical question. Where does a machine like this actually begin?
You have to start with the raw material.
Right. Before we can filter any data, before we can hunt for that perfect RA drug target, we need a massive starting point. So we arrive at our first major hurdle. Let's call it the problem of noise.
The notes explicitly specify that the foundation of this entire process relies on GWAS summary statistics.
GWAS, which stands for Genome-Wide Association Studies.
Yeah, and to understand the scale here, you have to realize that you cannot do modern causal genetics without immense continent-spanning statistical power.
The numbers in the notes are genuinely hard to wrap your head around. The pipeline pulls starting data from the UK Biobank, which is 500,000 individuals.
Right.
And then it pulls from FinnGen, which is another 500,000 people from Finland. And on top of that, it layers in data from the OpenTargets database.
So we're talking about mapping the genetic codes and medical histories of over 1 million distinct human beings just to get our starting lineup.
It's wild. Let's visualize what taking a genetic snapshot of 1 million people actually means.
Well, we aren't just looking at a few isolated genes. A GWS scans millions of distinct markers all across the human genome.
It's looking for correlations, basically.
Exactly. It asks a really simple statistical question. Do people with rheumatoid arthritis have a specific genetic variation more often than people who don't have the disease.
You know, I always think of a GWAS like taking a satellite photo of a massive continent at night.
Oh, I like that.
Yeah, you're looking down from space and you can see where the massive storms are happening. Those storms are the disease signals for RA.
You see a huge hurricane of genetic activity in one region.
Right, a thunderstorm over there. But from space, you can't see the individual houses taking damage. You just see the broad weather pattern.
There's a perfect analogy. The GWS gives you the rough neighborhood, like the zip code of the disease.
But having a million genomes doesn't mean the data is clean, does it?
Not at all. That's why the pipeline dictates extremely strict quality control, or QC, right at the start.
It applies brutal mathematical filters before the real analysis even begins.
Yeah, garbage in equals garbage out.
Well, I am looking at those exact filters right now, and they seem incredibly demanding. The notes dictate that we have to filter for a minor allele frequency, or MAF, greater than 0.01.
Right, 1%.
We also need an imputation accuracy, measured as an R-squared value greater than 0.8. And most surprisingly, we have to completely exclude an entire section of our DNA called the MHC region.
Which is a huge chunk of data to throw out.
Why are we throwing away so much data right at the start? Let's start with that minor allele frequency of 0.01.
Well, it all comes down to preventing the mathematical machinery from tearing itself apart later on.
How so?
Minor allele frequency simply means how common a specific genetic variation is in the general population. A MAF greater than 0.01 means we are only going to look at genetic variants that naturally occur in at least 1% of the people we sampled.
But 1% sounds tiny. I mean, why is that the floor?
In genetics, 1% is actually a very solid, highly visible footprint. Remember, human biology is terribly messy.
That's true.
If we try to track a genetic variation that only appears in 1 out of every 10,000 people, our statistical tools just start to struggle.
They can't find it.
They can't tell the difference between a genuine biological signal causing RA and just random background noise. It's like trying to listen to a single person whispering in the middle of a hurricane.
So by setting that 1% floor, we ensure we only track signals loud enough to be reliably measured across a million people.
Exactly. You can't analyze what you can't clearly hear.
Okay, that makes perfect sense. But what about this imputation accuracy, the R-squared greater than 0.8? I mean, what are we actually imputing here?
This gets into the hidden economics of genetic research. Sequencing the entire, complete 3 billion letter DNA code of a million people is still, well, it's prohibitively expensive and slow.
So they don't sequence everything.
No, massive databases like the UK Biobank use a clever shortcut. They don't read every single letter. They just read specific, known landmarks across the genome.
It's like skipping through a book and only reading the first sentence of every paragraph.
That's exactly it. But because human genetics follows highly predictable patterns, because certain genes are almost always inherited together, we can use advanced algorithms to guess the rest.
To impute the missing letters between the landmarks.
Precisely. We can fill in the rest of the paragraph with astonishing accuracy.
Ah, so the R-squared value is basically a grade on a math test. It's the confidence score of those statistical guesses.
Spot on. An R-squared greater than 0.8 means our mathematical confidence that we filled in the blanks correctly is 80% or higher.
We are demanding high-definition data.
Exactly. If the statistical guess is blurry, if the confidence drops to 50 or 60%, this pipeline ruthlessly deletes that variant from the study.
It just refuses to build a billion-dollar drug target on a bad guess.
Right. Too risky.
Which brings us to the most counterintuitive rule in this entire first step. The exclusion of the major histocompatibility complex, or the MHC region.
Oh, this one always trips people up.
For anyone familiar with biology, the MHC region is essentially the central command center for the human immune system.
It is.
And since our running example, rheumatoid arthritis, is literally an immune system disease, ignoring the MHC region feels like, well, it feels like investigating a bank robbery and refusing to interview the bank manager.
That's a great way to put it.
Why on earth are we throwing that specific data out?
It is a brilliant question, and it really highlights the difference between human intuition and algorithmic reality.
Okay, how so?
You are entirely right that the MHC region is deeply, fundamentally involved in RA. But genetically speaking, the DNA in the MHC region is a hyper-dense, incredibly tangled jungle.
A jungle?
Yes, it is constantly mutating, rearranging, and shuffling itself just to keep up with new viruses and bacteria.
So it's structurally chaotic.
Beyond chaotic.
Wow.
The genetic variants in the MAC region are so tightly linked together that standard statistical tools completely break down when they try to map it.
The math just fails.
If you feed the MHC region into this automated pipeline, the math will panic. It will generate thousands of false positives.
Oh, I see.
It'll tell you that every single house in that specific neighborhood is causing the disease, simply because the houses are built too close together for the satellite to tell them apart.
So to keep our data pristine, to ensure the automated causality engine functions perfectly, we just exclude the jungle entirely.
We have to. We hunt for the hidden, actionable signals outside of the MHC.
We really have to clear the static. Okay, so stepping back, our step one foundation is set. We have a high-definition, highly controlled, million-person satellite map of rheumatoid arthritis signals.
But a satellite map of a neighborhood isn't enough to invent a cure.
No, we need a specific street address. We have to solve what we'll call the problem of the neighborhood.
Right. This represents the transition from broad genetic association to surgical fine mapping. We know the rough zip code of the disease. Now we have to pinpoint the exact specific typo in the DNA that's driving the signal.
And the note specified the tool used here is pulled from the SuSiE software stack. Specifically, it's a function called SuSiE.
Yes, step 2.
It takes our massive summary stats from step 1, combines them with an LD reference, which stands for linkage disequilibrium, and outputs things called credible sets and posterior inclusion probabilities, or PIs.
Let's demystify that jargon. Starting with linkage disequilibrium, or LD. It sounds intimidating, but it is really just the biological version of a poorly shuffled deck of cards.
A poorly shuffled deck. Elaborate on that.
Well, when human beings reproduce, our DNA gets shuffled before it's passed to the next generation. But it doesn't get shuffled perfectly.
It sticks together.
Exactly. Chunks of DNA that sit right next to each other on a chromosome tend to stick together. They get inherited as a single unbroken block. That block is in linkage disequilibrium.
So if variant A is the actual genetic typo causing RA, but variant B and variant C happen to live right next door on the same block of DNA.
They all get inherited together, yes.
And because they are inherited together, your step 1 GWAS satellite photo will show all three of them lighting up like a Christmas tree.
GWAS will say, look, variants A, B, and C are all highly correlated with the disease.
But biology tells us that usually only one of them is the actual culprit.
Exactly. The others are just innocent bystanders who happen to catch a ride on the same genetic block.
I see. And that leads to a crucial observation from the source text. The notes are extremely explicit that this pipeline relies on a brand new tool called SuSiE 2.0.
It's a huge upgrade.
It specifically warns against using the older, traditional method known as LD clumping. Why is SuSiE 2.0 such a paradigm shift? What was fundamentally broken about the old clumping method?
Let's use our neighborhood analogy again. The old method, LD clumping, basically draws a big red circle around that whole genetic neighborhood.
Okay.
It looks at the group of variants A, B, and C and says, well, variant A is screaming the loudest on the statistical readouts, so we will crown variant A as the sole culprit and just ignore everything else in the circle.
It just assumes there's only one bad actor per neighborhood.
Exactly. It completely forces a single answer. But human biology is deeply complex.
Right. There could be more than one.
What if there isn't just one culprit? What if, in this specific neighborhood, variant A causes rheumatoid arthritis by messing with a protein structure, but completely independently, variant F on the other side of the neighborhood also causes RA by entirely different means?
LD clumping would entirely miss variant F. It would just lump it into the background noise because variant A was louder.
Precisely. We would lose a potentially massive drug target. But SuSiE, which stands for Sum of Single Effects, was mathematically designed from the ground up to solve this exact problem.
It untangles them.
Yeah, it's an algorithm capable of untangling regions where multiple, totally independent genetic variants are contributing to the disease simultaneously.
So it can look at the chaotic neighborhood and mathematically isolate the signals. It can say, we have a culprit at house A, and independently we have a second culprit at house F.
The precision there is incredible. And the notes say SuSiE outputs these results as credible sets and PIPs.
Okay, let's break those down. What is a credible set?
A credible set is exactly what it sounds like. It is a highly refined, short list of genetic variants.
Like a suspect list.
Exactly. The algorithm is telling us, I have mathematically eliminated the background noise, and I am highly confident that the true causal variant is somewhere on this short list.
And the PIP, the posterior inclusion probability, is the score for the individual variants on that list, right?
Correct. It is a Bayesian probability score ranging from 0 to 1. If SuSiE gives a variant a PIP of 0.9, it means the algorithm is 90% confident that this specific singular typo is the one driving the disease signal.
But the source material doesn't stop there. There is a detail here that elevates this from just a great pipeline to a truly revolutionary one.
Oh, the MF SuSiE upgrade.
Yes, it dictates using something called mSuSiE to achieve cell type resolution.
This is where the pipeline separates itself from 99% of genetic research happening today.
Let's think about why that matters for our RA patient. We just used SuSiE to find the exact street address of the genetic typo, but our bodies are made of trillions of cells.
Trillions.
A neuron in your brain, a muscle cell in your bicep, and a white blood cell in your veins all contain the exact same DNA manual. They all contain the exact same typos.
But a brain cell reads completely different chapters of the manual than an immune cell does.
Right, which means a genetic typo only matters if it happens in a chapter that the cell is actually trying to read.
If our pipeline flags a variant for array, but that variant only seems to affect the biology of brain tissue, well, that's a massive red flag.
Yeah, RA is a disease of the joints and the immune system. We don't want to develop a drug targeting brain chemistry to fix a swollen knee.
That would be a disaster.
We want to know if this DNA typo is actually causing havoc in a macrophage or a T cell, the actual infantry of the human immune system.
That is the magic of MF SuSiE. It allows the computational pipeline to fine map these variants while simultaneously probing diverse biological data sets.
Cross-referencing.
Exactly. It asks, in which specific cellular environment is this genetic effect actually waking up and causing damage?
So if the algorithm proves that our RA variant is exclusively wreaking havoc inside immune cells, our confidence that we are tracking the true mechanism of the disease just skyrockets.
Exactly.
That is a phenomenal transition. So we've solved the problem of the neighborhood. We know exactly which house has the problem, and we know exactly what kind of cell lives in that house.
But that brings us to our next massive hurdle. Step 3.
Right. Let's call it the problem of the blueprint versus the factory. Knowing a typo exists in a T cell doesn't actually tell us what the typo is doing to the cell.
This is a conceptual leap that trips up a lot of people. Finding a variant linked to a disease doesn't tell you the mechanism of the disease.
Because it's just a location, right?
Right. Most of the genetic typos we find in these massive GWAS studies don't actually change the physical shape or structure of a protein.
Wait, if they don't break the protein, how do they cause the disease?
They act like dimmer switches. They sit outside the gene, in the regulatory regions of the DNA, and they turn the volume of a nearby gene up too high or down too low.
They alter gene expression.
Exactly.
And to figure out which dimmer switch controls which gene, the pipeline introduces an entirely new universe of data called eQTLs, or expression quantitative trait loci.
The nodes list sources for this data like GTX, eQTLGen, and very specifically a dataset called 1K1K.
And the mathematical tool used to blend this all together is called COLOC.SuSiE, armed with something called TSS priors. And the brutal filter we must pass here is a probability of H4PRH4 greater than 0.8.
That is a dense wall of terminology right there. Let's break it down piece by piece, starting with the goal. The goal is colocalization.
I want to try a forensic analogy here for colocalization.
Go for it.
Think of colocalization like a detective investigating two seemingly separate crimes in a major city. Crime number 1 is our genetic disease, rheumatoid arthritis. People are getting sick.
Okay, what's crime 2?
Crime number 2 is an altered gene expression. We found a specific gene whose volume has been cranked up to dangerous levels. Colocalization is the incredibly difficult forensic process of proving that both of these crimes were committed by the exact same suspect, our single genetic typo from the SuSiE step.
That is a highly accurate way to look at it.
Because, as we established with the neighborhood problem, two variants can sit right next to each other on the DNA strand. Variant A causes the disease. Variant B turns up the volume on gene X.
And because they are neighbors, they are inherited together.
Right. A lazy researcher might look at that and say, aha, gene X must be the cause of rheumatoid arthritis. But they are wrong. It's just a coincidence of geography.
It is the ultimate trap in genetics. Colocalization uses Bayesian statistics to test the hypothesis, and that is what the H4 stands for in PRH4.
A hypothesis 4.
Yes. Hypothesis 4 is the mathematical assertion that a single, identical, shared genetic variant is driving both the disease and the altered gene expression.
And we demand a Bayesian probability greater than 80% before we believe it.
We need that certainty.
But let's pause and unpack the data sources powering this math, because the source notes make a very specific distinction here. They mention older databases like GTEx, but they heavily emphasize that dataset called 1K1K, specifically noting it for single-cell eQTLs.
This is a huge point.
Why is the shift to single-cell data considered a revolution here?
It goes back to our discussion about the immune system. Older databases like GTEx were foundational, but they had a massive physical limitation. They relied on bulk tissue sequencing.
Bulk tissue, like testing a whole organ.
Sort of.
Mm.
Imagine taking a vial of whole blood or a chunk of liver tissue and throwing it into a blender. You grind it all up into a biological smoothie, and then you measure the average gene expression of the entire mixture.
But blood isn't just one thing. It's a soup of red blood cells, dozens of different types of white blood cells, platelets.
Exactly. If our RA typo is only acting like a dimmer switch in one incredibly rare, highly specific type of killer T cell, that tiny signal gets completely drowned out when you measure the average of the whole blended smoothie.
The background noise of the millions of other normal cells completely masks the disease signal.
It's invisible.
So how does 1K1K solve the smoothie problem?
Through microfluidics and single-cell sequencing. 1K1K doesn't blend the tissue. It literally separates the blood into thousands of microscopic droplets, trapping one individual cell in each droplet. It then sequences the gene expression of every single cell individually. It looks at every ingredient in the soup one by one.
That is mind-bending technology.
It changes everything. By feeding 1K1K data into the colocalization algorithm, this pipeline achieves absolute, irrefutable cell type resolution.
It doesn't just say this variant changes gene expression in the blood.
No, it says this specific typo cranks up the volume of gene X, but only when it is sitting inside a CD4 positive memory T cell.
It's like proving the suspect committed both crimes using the exact same highly specialized lockpick.
Exactly.
Now, I have to ask about one more piece of jargon in this step before we move on. The notes say that colocalization uses TSS priors. What does that mean?
TSS stands for Transcription Start Site. Think of it as the giant neon Start Reading Here sign that sits at the beginning of every gene on the DNA strand.
Okay, and the prior?
A prior is a concept from Bayesian statistics. It's a way of injecting known biological reality into raw math. We know biologically that if typo is going to act as a dimmer switch for a gene, it is usually located physically close to that neon start sign.
So a TSS prior is essentially telling the mathematical algorithm, hey, while you are hunting for the culprit, you should probably focus your search on the variants physically closest to the start of the gene.
Exactly. It makes the algorithm faster and vastly more accurate by pointing it in the biologically logical direction.
Okay, so let's tally up our victories. We have the specific typo. We know it causes RA. We know it alters the expression of a specific gene, and we know it does this inside a specific immune cell.
By most standards, that is a spectacular day in the lab.
It really is. But the notes say we aren't even close to done. This brings us to step 4, which we can call the problem of the assembly line, or MOLOC triangulation.
Because biology is deeply, fundamentally redundant and prone to coincidence. Two overlapping signals, the disease and the RNA gene expression, are strong evidence.
But not strong enough.
If you were going to tell a pharmaceutical CEO to spend $1 billion designing a chemical to attack this target, two signals are not enough. We want to triangulate the truth across the entire biological assembly line.
The pipeline deploys a statistical tool called MOLOC to integrate a third trait into our forensic investigation.
Step 4.
Right. We take our GWAS data, the disease, and our eQTL data, the RNA blueprint, and we combine it with either mQTL or pQTL data.
The notes specify that mQTL represents methylation data from a source called God-DMC.
And pQTL represents protein data from UKB-PPP or decode. And the filter we must survive here is a PRGEM score greater than 0.8, which signifies full 3-trait sharing.
We are adding entirely new, massive dimensions of biology into the math.
Let's walk through the assembly line, because this is where the sheer elegance of the pipeline shines. We aren't just looking at DNA and RNA anymore.
No, we are looking at the central dogma of biology. DNA makes RNA, and RNA makes proteins.
It is the factory floor of human life. DNA is the master blueprint locked in the vault. RNA is the photocopied instructions sent down to the factory floor.
And proteins are the actual physical machines, the gears and levers, built from those instructions.
So up until step 3, we had only proven that a typo in the master blueprint was linked to a misprint in the photocopied RNA instructions.
Right. But sometimes the human body is incredibly smart. There are proofreading mechanisms.
Like quality control.
Exactly. Sometimes the body sees the misprinted RNA instructions and says, wait, this is wrong, and it stops the assembly line before the faulty protein machine is ever built.
Oh. And if that happens and we design a drug to attack a protein machine that doesn't actually exist, well, the drug fails.
Utterly fails. That is why we bring in pQTL's protein quantitative trait loci. We query databases like the UK Biobank Pharmaproteomics Project, the UKBPPP.
Which measures the actual physical circulating protein levels in the blood of tens of thousands of people.
We are checking the end of the assembly line. We are verifying that the faulty machinery actually got built and pushed out into the bloodstream.
That perfectly explains the pQTLs, the proteins. What about the mQTLs? The notes mention methylation data from GWAS. What is methylation?
Methylation is the science of epigenetics. It's the mechanism the body uses to turn genes on and off without actually changing the underlying DNA code.
How does it do that?
Think of it like a physical padlock clamped directly onto the DNA blueprint. If a gene has a lot of methylation, it's locked down tightly. The cell can't read it.
So an mQTL is a genetic typo that accidentally places a padlock where it shouldn't be or removes a padlock that should be there.
Exactly. By using MOLOC multiple trait colocalization, we are forcing the math to find a single shared genetic variant that controls three things simultaneously.
The triangulation.
Yes, it must affect rheumatoid arthritis risk,
Yeah.
and it must alter the RNA instructions, and it must alter the final protein levels or the physical DNA padlocks, all in perfect synchronization.
That is what the PRGM score of 0.8 means. It is the mathematical proof of that perfect three-way triangulation.
It's the ultimate alibi check.
It really is. It's like we caught them planning the crime on the DNA blueprint, we caught them talking about it on the RNA burner phones, and we literally found the physical protein weapon in their hand.
And when all three layers align, the false positive rate drops to near zero. It is no longer a statistical correlation. We are watching the gears of the disease turn in real time across the entire biological spectrum.
It feels like an insurmountable mountain of evidence. But astonishingly, step 5 is where the pipeline gets truly ruthless. Step 5 addresses the problem of coincidence.
The causality problem.
Yes, it is the pursuit of absolute cause and effect through Mendelian randomization.
Because here is the harsh, unforgiving truth of pharmacology. Even a perfect, beautiful three-way overlap across DNA, RNA, and proteins is technically still just a correlation.
And if your goal is to cure a disease, correlation is a mirage. Let me push back on that because it sounds extreme. We just proved that our typo changes the blueprint, builds a faulty protein, and is perfectly linked to the disease. How can that possibly be a mirage?
Let me give you a classic example that has burned pharmaceutical companies for billions of dollars. Let's say we find a perfect correlation between a specific inflammatory protein and rheumatoid arthritis.
Okay.
People with RA have incredibly high levels of this protein in their joints. The math is undeniable. A pharmaceutical company looks at that and thinks, fantastic, this protein is causing the swelling. We will engineer a drug that destroys this protein and we will cure the arthritis.
I mean, it sounds totally logical.
It does. Until they run the clinical trial and the patients actually get worse.
Worse, why?
Because they confused correlation with causation. What if the high protein levels weren't the cause of the RA? What if the rheumatoid arthritis was causing the high protein levels?
Oh, the direction was flipped?
Yes. What if that specific protein was actually the body's natural fire extinguisher, desperately being pumped into the joint to try and stop the autoimmune fire?
Oh, wow. If you design a drug to destroy that protein...
You just ripped the fire extinguisher out of the patient's hands. You actively fed the fire.
That's terrifying.
It is the nightmare scenario of drug development. To design a safe, effective drug, we must prove the direction of causality.
We have to prove mathematically that altering this specific biological target will actively change the disease outcome in a helpful way, not a harmful one.
Step 5 is the ultimate uncompromising test of that causality.
The notes detail the use of Mendelian randomization, or MR, leveraging the cell type-specific data we generated in earlier steps. And the rigor demanded here is intense.
It really is.
The pipeline requires researchers to use a minimum of 5 different MR statistical methods, pulled from 3 completely different assumption families. The list reads like a roll call. IVW, Meror-Egger, Messer-Epi-S, weighted median, Mr. Presso, and Mr. SPI.
We have to explicitly define the estimand, and we must pass a strict quality check where the F statistic is greater than 10.
It is a statistical gauntlet unlike anything else in biology. Let's start with the concept of Mendelian randomization itself. How does looking at genetics prove cause and effect?
Think of Mendelian randomization as nature's version of a multi-million-dollar randomized controlled clinical trial. When a human being is conceived, nature essentially flips a coin millions of times, randomly assigning you genetic variants from your parents.
Right.
Because these genetic assignments happen at conception, long before you ever develop a disease or change your lifestyle, we know that genetics came first.
The genetics precede the disease. Time only flows in one direction.
Exactly. We can use those randomly assigned genetic typos as instruments to pry open the truth.
How does the instrument work?
Well, if the people who are randomly assigned the typo that raises gene X expression always go on to develop higher rates of rheumatoid arthritis, we have proven causality. The disease cannot reach backward in time to change your DNA at conception.
Therefore, the gene expression must be causing the disease.
That's it.
That is profoundly elegant. But to make this work, the math has to be rock solid, which brings me to this F statistic rule. The notes say the F statistic must be greater than 10. Let me try translating this.
Go ahead.
Is the F statistic basically a measure of how strong our genetic instrument is?
No.
Like if we are trying to pry open a vault door to find the truth, we don't want a flimsy plastic pry bar that will bend and snap under the statistical pressure. We need a solid iron crowbar.
That is a brilliant way to frame it. The F statistic mathematically measures the strength of the relationship between our genetic typo and the actual change in the gene's expression.
So if it's too low,
If the F statistic is below 10, it means our typo only has a very weak, flimsy, inconsistent effect on the gene. If we try to use a weak, wobbly instrument to prove causality for a massive, complex disease like RA, the math falls apart.
we get biased, unreliable results.
Exactly. Demanding an F stat over 10 guarantees we have a heavy-duty, reliable lever to pull.
Okay, so we have an unbreakable iron crowbar. But that brings me to the most baffling part of the notes. Why do we need 5 entirely different mathematical methods to pull that lever?
Because every single statistical method ever invented has a blind spot.
The pipeline insists on IVW, MR Egger, Mr. Presso. Why isn't one perfectly good math equation enough to prove causality?
In Mendelian randomization, the greatest threat, the ultimate villain, is a biological phenomenon called horizontal pleiotropy.
Horizontal pleiotropy. I'm definitely going to need an explanation for that one.
It is a fancy academic term for a genetic variant that acts like a secret multitasker. We want a variant that affects gene X and through gene X causes the arthritis. That is a vertical causal pathway.
But what if it isn't vertical?
Right. What if our variant affects gene X, but it also secretly affects gene Y, and it's actually gene Y that is causing the disease?
The variant is secretly moonlighting. It frames in gene X for a crime actually committed by gene Y.
Yes. That secret horizontal pathway completely ruins our causal proof. And each of those NR methods you listed handles the threat of horizontal pleiotropy in a completely different way.
So they're like specialists.
Think of them as a team of 5 different homicide detectives, each with radically different personalities, investigating the same suspect.
Oh, I love this. Let's run with that. Who are these detectives?
First, you have IVW, or inverse variance weighted. IVW is the most statistically powerful detective, but he is blindly optimistic.
Optimistic in what way?
He assumes that horizontal pleiotropy simply doesn't exist. He assumes the suspect is telling the truth.
That sounds dangerous.
It is, which is why he works with MR Egger. MR Egger is the deeply cynical, paranoid detective. He assumes that every single genetic variant is a secret multitasker.
So he assumes everyone is lying.
Exactly. He mathematically calculates the bias, the intercept, and tries to subtract all the lies from the data before drawing a conclusion.
Okay, so we have the optimist and the cynic. Who else?
Then you have the weighted median method. This is the democratic detective. He doesn't trust any single piece of evidence. He gathers dozens of different genetic variants, lines them up, and takes a democratic vote.
Majority rules.
Yes. As long as at least 50% of the variants are telling the truth, he arrives at the correct causal answer, ignoring the liars.
And Mr. Presso. That's a great name, by the way.
Mr. Presso is the aggressive, outlier-hunting detective. He looks at the spread of data, chemically and mathematically identifies the specific individual genetic variants that are acting weird or breaking the rules of the group.
Let me guess, he kicks them out.
He violently kicks them out of the equation entirely before running the final numbers.
So the logic here is staggering. We aren't just running a test, we are running a tribunal.
Exactly. And the blindly optimistic detective, the paranoid cynic, the democratic consensus, and the aggressive outlier hunter all interrogate the massive data sets.
And they all agree.
And they all come to the exact same conclusion that gene X definitively causes rheumatoid arthritis. Then we have achieved incredibly robust proof of causality.
It's bulletproof.
It is mathematically almost impossible for a false positive, a pleiotropic multitasker, to successfully trick all 5 distinct algorithmic methods at once.
That is breathtaking. We have pushed thousands of potential genetic variants through an incredibly harsh gauntlet.
And eliminated almost all of them.
We started with the noise of 1 million people, zoomed into the exact street address, proved it overlapped with RNA blueprints and physical protein machines in specific immune cells, and then battered it with a tribunal of 5 different tests of absolute causality.
Which brings us to step 6. After the gauntlet, we have to sweep the floor, see what actually survived, and keep score.
This is the gene prioritization scoring step. The notes lay out an exact, unyielding 5-point scoring system. Let me walk through the grading rubric.
It's very strict.
Point 1. If a variant has a SuSiE PIP greater than 0.9, meaning we are 90% sure it's the exact causal typo, it gets 1 point.
Point 2. If it passes the colocalization test with a PRH4 over 0.8, proving it definitively affects gene expression, it gets 1 point.
Point 3. If it passes the MOLOC triangulation, proving it perfectly aligns across DNA, RNA, and protein traits, it gets 1 point.
Point 4. If it shows significant causality in 3 or more of those Mendelian randomization detective methods we just discussed, it gets 1 point.
And the final point. If there's concrete 1K evidence that all of this is happening in a disease-relevant specific cell type, like a memory T cell for RA, it gets the 5th point.
A maximum of 5 points. A perfect score.
And the ultimate goal of this entire pipeline, according to the source, is to identify genes that achieve a score of 4 or 5.
Those are the winners.
If a gene hits that threshold, it is officially designated as a Tier 1 target. This whole process reminds me of a terrifyingly strict VIP bouncer at an exclusive underground club.
A bouncer? How so?
You don't just get in because you look the part or because you know the owner. You need 5 completely separate, highly authenticated forms of ID. You need a passport, a driver's license, a retina scan, a fingerprint, and a cryptographic background check.
That's a very intense club.
If you miss 2 of those, the bouncer throws you back out onto the street.
I love the cold, hard logic of this scoring system because it addresses a fundamental flaw in how science has historically been done.
What flaw is that?
In academic science, there is a very human, very dangerous flaw. Researchers fall in love with their own hypotheses.
We want our favorite suspect to be guilty.
Exactly. Imagine a brilliant researcher who has spent 15 years of their life and millions in grant money studying one specific biological pathway related to rheumatoid arthritis.
They're deeply invested.
Deeply. When the genetic data finally comes in, it is incredibly tempting, almost a subconscious reflex, to squint at the data, to look at a weak signal and say, well, if you look at it in this light, or if we ignore this one test, maybe it's actually significant.
Human bias creeps in to save a life's work.
This automated 5-point algorithmic scoring system completely removes human emotion from the equation. A researcher cannot just champion their favorite RA gene into a billion-dollar clinical trial through force of personality.
The data has to speak for itself.
The genetic data has to survive the gauntlet and objectively, mathematically, earn its Tier 1 status. It forces absolute intellectual honesty at an industrial scale.
And that profound intellectual honesty brings us to the climax of the pipeline. Step 7, from the computer to the pharmacy shelves.
This is the moment of truth.
Let's imagine we found it. We have our Tier 1 target gene for our running example of RA. It survived the noise, the neighborhood, the assembly line, and the tribunal. It scored a perfect 5.
The holy grail.
The final step is answering the trillion-dollar question. Can we actually drug it?
This is drug target annotation. It is the exact moment where theoretical molecular biology slams into the realities of pharmacology.
The pipeline takes our pristine Tier 1 gene and aggressively queries massive global pharmacological databases. The notes list OpenTargets, DrugBank, and ChEMBL.
We are looking for any known chemical compound, any molecule that can physically bind to and interact with the protein our target gene creates.
And the text explicitly splits the outcomes of this search into two highly distinct buckets.
These two buckets represent wildly different futures for patients suffering from RA and wildly different financial realities for pharmaceutical companies.
Right. If the database search shows that an existing FDA-approved drug already acts on our target gene, it's categorized as an opportunity for repurposing.
But if the gene is mathematically druggable based on its physical protein structure, but no human being has ever bothered to invent a drug for it yet, it's categorized as a novel target.
Let's dive into the sheer real-world impact of those two buckets. The difference is staggering, isn't it?
It really is.
Yeah.
If our newly discovered RA gene falls into the novel target bucket, it means we have to design a drug from absolute scratch.
Which means we start at zero.
Less than zero. We have to invent a new chemical molecule. We have to test it in petri dishes. Then we test it in animal models to see if it destroys their liver.
And then human trials.
If it passes all that, we run phase 1 clinical trials in a few healthy humans just to prove it isn't toxic. Then phase 2 efficacy trials in a small group of RA patients. Then phase 3 massive multinational trials.
That sounds incredibly long.
We are talking about a grueling decade of work, easily costing over $1 billion.
And as we established at the beginning of this deep dive, historically, 90% of those novel drugs fail somewhere along that 10-year journey of misery.
Exactly. But the other bucket, repurposing, that is the absolute holy grail of modern medicine.
How so? Give me an example.
Let's say our pipeline finds a perfect causal tier 1 gene for rheumatoid arthritis. We query the DrugBank database, and the computer spits out a match. It turns out there is already a cheap, widely available, FDA-approved drug on the market that brilliantly targets this exact genetic mechanism.
But it's for something else entirely.
Yes. It's currently being prescribed by doctors to treat a rare type of asthma.
The implications of that are mind-boggling.
Because that asthma drug has already been on the market for 10 years. It has already passed phase 1 safety trials. We already know definitively that humans can swallow this pill for years without suffering toxic side effects.
You can literally skip half a decade of safety development.
You can almost immediately launch targeted clinical trials to see if this asthma pill cures rheumatoid arthritis. You save hundreds of millions of dollars, but vastly more importantly, you save years of time for patients who are suffering right now.
It's like finding out the cure for the disease was already sitting in your medicine cabinet. We just didn't know we were supposed to use it.
It's incredibly powerful.
But even if we are unlucky, even if we are forced to go down the long, hard road of developing a novel target, this 7-step pipeline fundamentally changes the math of drug development, doesn't it?
Oh, it changes the entire paradigm.
Yeah.
Let's go back to that 90% failure rate. Why do so many novel drugs fail in phase 3 trials after billions have been spent?
Because the initial target was wrong.
Most often, they fail for a tragic reason. The underlying biology was flawed from day one. The company was chasing a false positive. They targeted a gene that was correlated with the disease but didn't actually cause it.
Or they got the wrong cell type.
Or they targeted a gene that was truly causal, but it only caused the disease when active in a brain cell, and the drug they designed couldn't cross the blood-brain barrier.
But our Tier 1 target isn't a guess. It survived the Mendelian Randomization Causality Tribunal. It survived the 1K1K single-cell resolution tests.
We know exactly what it does and exactly which T cell it does it in.
Exactly. Because this specific 2026 pipeline guarantees cell type resolution and multi-omic causal evidence, pharmaceutical companies can look at these novel targets with a massively reduced fear of failure.
The biological risk, the terror of chasing a ghost, has been surgically removed by the software algorithms before a single human chemist ever picks up a test tube.
The probability of that novel drug succeeding in phase 3 trials must go way up.
It doubles, triples, or even quadruples?
I have to admit, I am completely captivated by the scale of this. We have mapped the entire gauntlet. But I want to spend a moment looking under the hood in Section 8, the engine room and publishability.
It's definitely worth looking at the logistics.
Now that we understand the profound depth of the science, the Bayesian math, the single-cell microfluidics, the causal tribunals, let's look at the sheer logistics of running this beast. Why do the notes explicitly claim this specific setup is the absolute bleeding edge of science in 2026?
The computational efficiency of what the authors have engineered here is almost difficult to believe.
I am looking at the compute times in the text right now. Listen to this. For every single genetic locus, every single neighborhood we investigate, it takes the software only 5 to 30 seconds to run through an individual step of this pipeline.
Which is blazing fast.
If we have 2,000 potential loci for disease like RA, running this entire massive multi-omic analysis sequentially on a standard single computer core takes about 30 to 50 hours.
Which, frankly, is already incredibly fast, considering the sheer volume of Bayesian probability math being calculated at the single-cell level.
But it gets so much better. Because the pipeline is fully automated and modular, it can be parallelized.
You can split the work up.
If you distribute this monumental computational task across a modern server equipped with 100 computer cores, the entire process from the noisy GW all the way to the pharmacy shelf drops from 50 hours down to just 30 to 60 minutes.
1 hour.
1 hour. And the source notes are very explicit about why this specific configuration is highly publishable, why it is so rare to see it all in one place.
It lists all the required elements.
It does. It lists the use of SuSiE 2.0 instead of the outdated clumping, the use of variant-specific TSS priors, the MOLOC multi-omic triangulation combining RNA and proteins,
and the 1K single-cell resolution.
and the explicitly defined Mendelian randomization estimands fighting pleiotropy, and the systematic bias-free 5-point scoring.
I want to ask you a question. Looking at that massive list of requirements, what stands out to you the most?
What stands out to me is the sheer, overwhelming number of disparate fields of expertise required to build this machine. To create this pipeline, you need statistical geneticists, molecular biologists, pharmacologists, high-performance software engineers, and big data architects.
And that leads directly to the final, most striking fact from the source material. As of early 2026, the authors state that only about 5 scientific papers globally have successfully combined all of these elements into a single, cohesive, end-to-end analysis.
Only 5. Wow.
It is incredibly difficult to execute this correctly.
Because most academic labs are highly specialized silos, right?
Precisely. You have labs that are world-class experts in GWAS, but they don't do single-cell. You have labs that are brilliant at Mendelian randomization, but they don't understand protein triangulation.
Stitching all of these wildly complex tools together without the code breaking?
Without the subtle statistical assumptions of one algorithm violating the mathematical rules of the next, it is an absolute masterclass in modern computational biology.
Let me summarize the epic journey we've just been on. We started with massive, chaotic crowds of data, 1 million people in a GWS acting like a blurry, noisy satellite image.
The noise.
We tackled the problem of the neighborhood, zooming into the exact street address using SuSiE 2.0 fine mapping. We tackled the problem of the blueprint, using colocalization and single-cell data to prove that our genetic suspect was at the scene of the RNA crime in the exact right T-cell.
Then the assembly line.
We conquered the assembly line with OLOC triangulation, tracking the biological cascade from DNA to RNA to physical protein machines. We locked down the alibi by proving absolute cause and effect with a tribunal of 5 different Mendelian randomization detectives.
And then the banser.
We stripped away human ego and bias by enforcing an objective 5-point VIP bouncer score. And finally, we scanned global pharmacological databases to find out if our surviving Tier 1 targets could be repurposed immediately to help patients suffering from rheumatoid arthritis.
It is a perfect distillation of a paradigm shift. We are moving away from the era of artisanal, bespoke, one gene at a time guesswork, and we are entering the era of industrialized, automated biological truth-seeking.
Which leaves me with a final, highly provocative thought to mull over, and I want you, the listener, to think about this deeply as well.
What's your takeaway?
We started this deep dive talking about finding a single misprinted letter in 1 billion books. We talked about how long it takes human chemists to guess the right answer. But this pipeline is like inventing a massive automated search engine for biological causality.
It really is. It sees straight through the noise.
And if a 100-core server can map out irrefutable, Tier 1 causal genetic targets for incredibly complex diseases in just 60 minutes.
It's staggering to think about.
If the software can hand us the perfect, flawless blueprints to the exact broken machinery inside our cells that fast.
It raises a staggering question about the future. How long until the biggest bottleneck in human health isn't discovering what causes a disease?
But physically manufacturing the cures fast enough to keep up with the software? We are rapidly approaching a reality where we will have a massive backlog of absolute biological truth just sitting on servers waiting for the human chemist to catch up.
That is a wild thought to end on.
Thank you for joining us on this incredible deep dive into the 2026 causal genetics pipeline. I encourage you to look at whatever messy, complex data you are analyzing in your own life or career through this incredibly rigorous multi-step lens. Don't settle for the mirage of correlation, build a gauntlet, and demand the causal truth.